Micro-dystrophin gene therapy is a way of replacing the dystrophin gene in patients with Duchenne muscular dystrophy. The dystrophin gene is the largest gene in the genome, which presents enormous challenges, and it is impossible to package the entire gene into a virus for gene therapy because of its size. But over the last 15 or 20 years, scientists have been able to define which are the most essential parts of the gene for correcting the defect in Duchenne, and have been able as a result to design smaller and smaller and smaller versions of the dystrophin gene, which have been called mini-dystrophins and micro-dystrophins, that are able to compensate for most of the dystrophin function without replacing the entire gene. So micro-dystrophin gene therapy is a way of compensating for the function of dystrophin without replacing the entire gene, but rather a much smaller version that can be easily packaged into a virus. Viral gene therapy for Duchenne presents a number of really big challenges. The first one is, how do you package a gene the size of dystrophin? And the answer to that is to package smaller versions of the dystrophin gene, which are called microdystrophins, smaller and smaller versions which have the essential functions. A second challenge is not just that dystrophin is the largest gene, but it needs to be replaced almost throughout the entire body. So, in theory, all muscle groups, including muscles for breathing, the heart muscle, the brain, and other muscles what we call smooth muscles, for example, in the gut. All of these muscles and tissues use dystrophin routinely, and therefore, to really fully correct the problem in Duchenne, the gene would need to be replaced in all these tissues. So as a result, the virus will need to deliver the gene to all of these tissues, which means that you need a lot of virus. And so this is probably the major challenge, because it presents problems such as how do you make so much virus? So there are manufacturing challenges in order to prepare enough viruses for gene therapy treatment, but also because a lot of virus needs to be used for each patient. This presents challenges to the patient in terms of safety, because you're not giving the patient a small amount of virus, you're giving the patient a large amount of virus. And although these are not live viruses, what they do is stimulate the patient's immune system. And so there is a risk that there will be an immune response or an immune reaction from patients who are treated to these viruses. Now, typically, there are ways that you can overcome this, for example, by having some kind of temporary immunosuppressive treatment while the gene therapy is being conducted. So, for example, steroids or other types of immunosuppressive drugs could be used. But the real challenge comes should you need to give the virus treatment a second time. This would be expected to be difficult, because if the immune system has already been made aware of the virus the first time, it's likely that it will be much more reactive the second time. So the challenges are really the size of the dystrophin, delivery to virtually the entire body, the entire patient, and then the consequence of that for the amount of virus, manufacturing, and delivery of a large amount of virus into the patient, which then has potential immune or problems which prevent re-administration of the virus a second time. Exosomes are naturally occurring nanoparticles. So nanoparticles are, by definition, very small particles. And exosomes are a type of natural nanoparticle. So when most people talk about nanoparticles or nanotechnology, they typically think about things you make in the lab, synthetic nanoparticles. Exosomes exist naturally. And exosomes are small nanoparticles whose natural function is to convey messages between cells in the body. We didn't know about these until 30 years ago, and we've only just discovered within the last 10 years what exosomes do, which is to convey messages between cells in tissue. So, for example, between cells in a muscle, but even between cells in different tissues. So for example, between the muscle and the heart, or between the muscle and the brain. So these are natural. They typically contain natural messages to regulate how cells interact or behave towards one another. But because they exist and we now understand a lot about how they work, it is becoming possible now to utilize these as an alternative way of delivering drugs and medicines into cells in the body. So exosomes work as a natural communication system within the body of complex multicellular creatures like ourselves, but all mammals would have similar exosome systems. So these exosomes or nanoparticles are produced by cells. They are released from cells containing information from the cell. So for example, from a muscle cell, there would be information packaged into an exosome, and the exosome as a little particle, a little package of information, is released from the cell. It will then travel typically through the fluid outside cells or through the blood to other cells, where the exosome is then taken up, internalized into the recipient cell, and that information is then decoded and gives information to the cell that receives the message. And typically these kinds of messages might relate to regulating the way that cells work. So for example, regulating stem cells within tissue, or regulating your immune system, or regulating your blood cells. In other words, it brings a way of coordinating actions and activities of many cells in a complex organism. It brings a coordinated, controlled way to doing that. The unique thing about exosomes is not just that these are nanoparticles that travel between cells, but in contrast to the other types of communication systems in the body, so for example hormones or nerves, exosomes contain many messages. So they are particles that are packed full of messages. So it's not just a single message that goes from cell to cell, it's a complicated collection of messages. In other words, that message has a complex information that will allow the cell that receives it to do many things. So it's not a single message, not a single letter, not a single instruction, but a complex set of instructions that can then be decoded by the cell that receives the message and translated into a complicated series of actions, which might be telling the cell to divide or to grow or to react to a foreign virus or something like that. So complicated actions to coordinate the way that cells behave. So exosome therapies should be highly beneficial for Duchenne because of the possibility of targeting these specifically to muscles and heart, which is where the drugs are needed, and packaging complicated drugs into the exosome, such as genetic drugs, micro-dystrophin, oligonucleotides, all of which will be possible because this is the natural function of exosomes and how they work in the body. So one of the advantages of exosomes as a way of delivering drugs to Duchenne patients and other patients is that these are natural particles. Therefore, it is highly likely that they will be well tolerated by the immune system. They do not have foreign molecules, no viral molecules, no bacterial molecules. These are entirely human particles, and therefore it's very unlikely the immune system will see these as foreign and mount a damaging reaction. So this could be a very, very positive step forward for Duchenne therapy. So the potential of using exosomes as a way of delivering drugs has been developed over the last 10 years. And two of the crucial elements to doing that are, firstly, the ability to put the desired drug into the exosome, the genetic drug, and secondly, the ability to modify the exosome in such a way that it is targeted to a particular tissue, for example, the muscle. And over the course of the last 10 years, The technologies allowing us to do these two important things were developed in my lab here in Oxford and in the lab of my colleague Sami Andalusi in Stockholm. As a result of developing those technologies, we were then able, with the intellectual property that resulted, to establish a small company to develop this technology for therapeutic application. This company, called Evox Therapeutics, was founded in 2016, so 2 years ago, and has been working hard over the last 2 years to develop these technologies for therapeutic application in an industrial biotechnology setting. Evox now exists in Oxford, has a research lab and a head office in Oxford, employs up to 30 people currently, and is developing all of the technology and intellectual property to allow the development of new treatments for diseases like Duchenne. So Evox now has the chance, with Duchenne UK's support, to develop exosome-based treatments for Duchenne. Over the last 10 years, what has been really important is developing the technology allowing us to use exosomes for the delivery of therapies and genetic therapies to patients. In other words, to put the drugs in the exosomes and to target these to particular tissues. But as you can see, this is almost a technology that could be used for any disease. And Evox has many options and many possibilities for diseases it could develop treatments for. And it is therefore only with Duchenne UK's involvement and support that Evox can now concentrate a large effort on developing exosomes as a therapy for Duchenne, rather than concentrating its efforts on many, many other diseases for which the technology would be entirely applicable. I think Duchenne UK is obviously very important because it has not just been able to help Evox to understand what would be valuable for Duchenne patients in terms of new types of therapies, but has also been able to bring some funding support to allow the preliminary data to be generated using this technology. So as a result, Evox, in collaboration with Duchenne UK, has been able to develop a research project that will test this idea that exosomes could be developed as a therapy for Duchenne, and will generate the data over the course of the next 12 to 18 months to prove this.